We've treated over 1,500 patients, and I can tell you the clinical response is reproducible. Reproducible clinical effect. Our nurses see it, our residents see it, our patients see it. It's very uncommon for a patient to die. They die of sepsis, there's usually some underlying condition that we can't get source control or some other extenuating circumstance. And again, no side effects.
The eye mask protocol is based on ivermectin, the MATH plus on methylpred. However, there's already a lot of information, so I would suggest if people are interested, they can go to our website, the FLCC Alliance at flcc.net, and we'll be able to download a whole host of useful information. To state that we're facing a global catastrophe is perhaps an understatement. We've just passed 400,000 deaths in the US, approximately 250,000 cases a day, which exceeds that of many countries throughout this whole pandemic. What's important to recognize is the difference between SARS, MERS, and SARS-1. So SARS-CoV-2 in blue, you can see it reaches a highest concentration prior to the onset of symptoms. And so that's most infectious, so it reaches the highest concentration in patients are most infectious prior to the onset of symptoms. And this contrasts with MERS and SARS-1, making this a particularly infectious disease. So in order to treat this disease, it's absolutely essential to recognize the phases of COVID. And this has not been well discussed or well popularized. So the patients go through a number of phases. There's the incubation period, which lasts up to about 5 days. As we know, about 20 to 30% of patients remain asymptomatic and don't develop the symptomatic phase. Patients then move into the symptomatic phase. Again, viral replication is greatest prior to the onset of symptoms. However, as patients progress through the symptomatic phase, viral replication tails off. Patients then move into the early and late pulmonary phase, where you have no longer viral replication. So this is not a viral cytopathic effect, but it's due to the host response to viral debris. This is immune dysregulation with abnormalities of innate and adaptive immunity. Below, one can see the clinical symptoms, the symptomatic phase, fever, malaise, cough, diarrhea, confusion in elderly people. As they move into the pulmonary phase, shortness of breath, and then progressive hypoxemia. Again, this is an important slide. Basically looks at the phases again of this disease. You see, this is the phase of active viral replication. Patients remain infectious only during this period. However, what's notable is that the PCR remains positive for a much longer period of time after viruses stop replicating. So the presence of a positive PCR does not mean infectivity. You can see this is the period of PCR positivity. Then as the virus disappears, one gets the presence of both IgG and IgM antibodies, which really start together, which is somewhat unique. And the disappearance of the viral replication occurs at the same time as one gets adaptive immunity. So we see PCR positive for a period of up to 3 weeks, even though the patient may no longer be infectious. In terms of the phases, again, this looks at symptoms, with day 1 being the onset of symptoms. So generally, people have fever for about 12 days. They have cough for about 19 days. But you can see there is this transition period where patients become symptomatic, but do not have pulmonary signs, and often can get better and then get worse. So the onset of shortness of breath and pulmonary signs is usually at about day 7 to 8. 2 or 3 days later, they get admitted to the ICU, get onto mechanical ventilation. The lower hatched bars represent patients who die. So in terms of management of COVID-19, this is an important table. So this is based on randomized controlled trials looking at treatment which has succeeded and those which have failed. So those that have shown benefit in randomized controlled trials are in green. Those that are harmful are in red. Those that are of no or marginal benefit in brown or yellow. And we've divided this into pre-exposure, post-exposure, the symptomatic phase, and the pulmonary phase. So going through the major drugs which have been tested, hydroxychloroquine really remains of unclear benefit in the early phase. Remdesivir, basically in the early pulmonary phase, as we'll see, reduced time to recovery, possibly no mortality benefit. Lopinavir, ritonavir, of no benefit. Interferons may be of benefit inhaled early. It's harmful late. Tocilizumab, really of unclear benefit, despite the recent study, and appears it may only be of benefit in patients who are inappropriately treated with corticosteroids. Convalescent serum, as we'll see, have absolutely no benefit. And really, it's a mistaken concept that antibodies would work against a disease which is not blood-borne, and it defies understanding of mucosal immunity. In terms of monoclonal antibodies, the same really applies. We know that the Lilly and study, as well as the other study, was terminated early because of, the Regeneron was terminated early because of harm, increased risk of death in the hospitalized patients. It's also worth noting that the U.S. government, at the time that the studies were halted, spent $3 billion, that's with a B, dollars to stockpile vials of this medication, which was shown to be harmful. That $3 billion could have bought enough ivermectin to prophylax the entire country. Obviously, when a drug doesn't work, you try to find another indication. They try using this drug in the symptomatic phase. Its benefit is unclear. In terms of corticosteroids, as we'll go through, we know it's beneficial in the pulmonary phase. It tends to harm in the symptomatic phase. Looking at ivermectin, as we'll see, this is a truly astonishing drug. It was designed for COVID-19. It's been shown to be of benefit in randomized controlled trials in pre- and post-exposure prophylaxis in the symptomatic phase and in the pulmonary phase. So this table really should inform us on how to treat COVID-19. So again, this is the adapted table with the different phases, looking at the treatment modalities. Obviously, when a virus is replicating, you use antiviral therapy. It's obviously pointless giving antiviral therapy once the virus is dead. Once something is dead, it's dead. You can't kill something that's already dead. And trying to kill something that's dead is a futile exercise. Yet people persist in giving antiviral therapy in this phase, which is characterized by anti-inflammatory therapy. So this is methylprednisolone, enoxaparin, aspirin, ivermectin, and this combination of flavonoids, zinc, vitamin D, vitamin C, and melatonin, which we'll touch on briefly. What is the MATH protocol? Well, the most important component is intravenous methylprednisolone, of which we'll talk about, high-dose intravenous ascorbic acid, thiamine, and heparin. In addition, we add the plus part, ivermectin, statin, zinc, vitamin D, famotidine, and melatonin. More recently, in patients who are critically ill and responding really poorly, we recommend really high megadose vitamin C. So this is a study. So most subsequent studies actually use methylprednisolone rather than dexamethasone. And if you look at the number needed to treat to save one life, significantly better with methylprednisolone than with dexamethasone or hydrocortisone. Pulmonologists are really familiar with methylprednisolone. It's the drug of choice for organizing pneumonia. So, we initially, our focus was in the treatment in the ICU and the development of the MATH Plus, but we recognized that really what our duty and obligation is to control this pandemic is to prevent the disease and to treat the disease early. So hence we have pre-exposure, post-exposure, and early treatment. And if we want to at all control this pandemic, we need to consider prophylaxis and early treatment. Once the patients reach the ICU, we know that they're in big trouble. So this is the comments that we get every single day. We get hundreds of similar emails. My doctor told me there was absolutely nothing she could prescribe for me once I was diagnosed with COVID, and that if my lips turned blue or I could not breathe, to go to an ER. It's truly astonishing that this is the state of play currently in our country. We have no way of preventing, we do not have any accepted, recognized protocol for the prevention and treatment of early COVID. This is truly astonishing. We then need to move to ivermectin. So William Campbell and Satoshi Omura were awarded the Nobel Prize in 2015 for their 1975 discovery of ivermectin. It was first used in humans in 1987. In vertebrates, it binds to the glutamate-gated chloride channel. It then paralyzes the worm or the parasite. It's a very highly lipophilic drug. It's rapidly absorbed, has a plasma half-life about 16 to 28 hours. What's important to recognize is that ivermectin is on the World Health Organization's list of essential medications. Over the last 3 decades, approximately 3.7 billion doses have been distributed. That's a B. 3.7 billion doses of ivermectin have been distributed. It's a drug with broad-spectrum antiparasitic and remarkably antiviral activity. In addition, it has potent anti-inflammatory and immune modulating effects. It seems to be a drug designed specifically for COVID-19. Now, despite what's on the FDA website, which is completely distortion of the facts, it is a safe and well-tolerated up to 10 times the recommended dose. Again, looking at mortality, these are randomized studies, these are observational studies, a highly significant reduction in mortality. There's no other drug which is available to treat SARS-CoV-2 which has such an impressive effect on viral clearance, improvement of symptoms, and mortality. And it's truly astonishing that the NIH cannot make a decision and can neither provide recommendation for or against a drug which is safe, cheap. So the cost to the WHO of a 12 milligram tablet of ivermectin is 2 cents. 2 cents compared to over $3,000 for remdesivir, which has minimal benefit. There are actually 56 trials in all, so within a few weeks or months, we should have a much larger database. In addition to its effect in patients with established disease, ivermectin has remarkable activity in preventing SARS-CoV-2. So this is prophylaxis in healthcare workers, 788 randomized to ivermectin, 400 control, 0. That's 0 patients who got ivermectin developed SARS-CoV-2. This is a look at all these studies, both observational and randomized controlled trials, again demonstrating that ivermectin is a potent drug in both pre-exposure and post-exposure prophylaxis. In terms of its safety, this was a review of 25 years. Basically, they concluded that the drug has negligible adverse reactions in humans. It has a robust safety profile. It can be used in children as young as 6 months of age. Most of the reactions are due to the death of the microfilaria rather than the drug itself. In patients with lower, they rarely have an encephalopathy. It's not recommended in pregnancy or lactation. The only caution with ivermectin, it's a remarkably safe drug, is drug-drug interactions, most notably with calcineurin inhibitors. There is also a reaction with azole antifungal drugs. And obviously vaccination is important so that these measures do not compete with each other. They all complement each other. It's going to take months, if not years, to vaccinate the entire world population. So we're facing a catastrophe right now. Vaccination will not solve this problem. We need to institute other measures right now. There is also the problem of mutation. There are a number of mutant strains with this mutation in the spike protein. It seems that the variant found in South Africa appears to be resistant to neutralizing antibodies. So that may pose a problem, whereas other measures such as ivermectin will still remain effective. And the question of ivermectin is just so frustrating. We managed to get the NIH to reverse their previous stance on recommending against. However, their recommendations, neither for nor against, are so inadequate, inopportune, ignorant, that we've actually written a letter to them saying they clearly do not understand what they're talking about. So we're pleading to them to please change what they're doing. It just makes no sense. This is a drug which has been used for 30 years, has a remarkable safety record, is on the WHO list of essential medications, has been shown in randomized controlled trials to reduce the risk of death. However, because these are not large mega trials done in the USA or the UK, they have the xenophobic attitude that somehow these physicians all across the world have conspired to make up this data. So, viral clearance and mortality are hard endpoints. And it seems remarkable that all these clinicians across the globe would somehow conspire to make up this data. It's remarkable that we're facing this situation. Hopefully with the new Biden administration, things will start moving in the right direction. But I think the amount of misinformation is truly astonishing. And, physicians aren't prepared to think out of the box. They follow these ridiculous guidelines. The problem with the recovery study was that they used one dose. And people think that that's the way you treat them. And clearly that's not the case. And obviously you raise a really important question. And, that's why we're trying to get this information out there.
You mentioned xenophobia, and I wonder if it's pharma's phobia of the 3-cent drug versus something more lucrative.
Yes. So, that's, yes. So that's obviously what's underlying this. And it's difficult to talk about it, that the U.S. could spend $3.7 billion stockpiling monoclonal antibodies that don't work. That we know all the scandals around remdesivir, which is still recommended by the NIH, $3,000 a dose. Yet you have a drug which you can buy for 2 cents, and they will not recommend for it. So obviously, people must make their own conclusions, but it's clear that there's a lot of mischief afoot, and it's clear who's responsible for this mischief. I'm more than happy for people to email me because I think, we need to get the message out there. We can't just sit back and do what we're told to do. That has failed. Doing what has failed up until now is not going to change this awful trajectory. We have to change what we're doing. And I think the key is early treatment at home and aggressive treatment in hospital.